Cell division, it has two types, mitosis and meiosis. In mitosis or equational division, parent cell divide into two daughter cells with exact copy of chromosomes. In meiosis, have of chromosome present in daughter cells. Let's see mitosis. It was first introduced by Walther Flaming in 1882. DNA replication is followed by chromosome segregation generated into genetically identical two daughter cells. Stages of M phase starts with karyokinesis and ends with cytokinesis. There are four subtypes: prophase, metaphase, anaphase, and telophase. In prophase, the diffused chromatin in interphase slowly condense into well-defined chromosomes. Each chromosome has two sister chromatids. They are held together at centromere by member of structural maintenance of chromosome, SMC, called cohesin. There is disappearance of nucleolus and disruption of nuclear envelope. MCDK phosphorylates subunits of nuclear pore complex which disassemble of nuclear pore complexes. MCDK phosphorylates nuclear lamina and there is breakdown of nuclear envelope. There is exception in yeast. There is no nuclear envelope breakdown while chromosome migrates, which is called closed mitosis. At the end of prophase, there is disassemble of cytoplasmic microtubules and formation of mitotic spindle. What is mitotic spindle? It is microtubules which is polarized tubulin polymer with minus end embedded in a spindle pole and plus end pointing outward from the pole. It has three types: a kinetochore microtubule, polar microtubule, and astral microtubule. Kinetochore microtubule attached to kinetochore on centromere with plus end. Polar microtubule does not interact with chromosome. Overlap with polar microtubule from opposite pole. Astral microtubule form aster, radiate outward from the centrosome toward the cortex of cell. What is kinetophore? It is a giant multilayered protein, assembles at centromere region of chromosome. It has three layers. Outer is interaction surface for spindle microtubules. Inner layer forms interface with chromatin. Central kinetophore is the region between outer and inner. Foundation for assembly of kinetochore is provided by centromere. Centromere is heterochromatic region with repeated sequence called alpha satellite DNA. 171 BP, holocentric chromosome is chromosome that lacks localized centromere and primary constriction, kinetochore diffuse attaching along the length of chromosome. Let's see metaphase. During this phase, spindle fibers assemble and disassemble, seeking attachment of chromosome. They are attached by two kinetochores on each chromosome, two sister chromatids, facing opposite direction by process called search and capture. Plus end of microtubules radiating out from MTOC attached to kinetochores or randomly hit and stabilize. Kinesin 4 and 10 is plus end directed protein. They push chromosome attached chromosomes from pole, interact with polar or astral microtubules, which is called polar ejection force. Dyneins are minus end motor proteins that link plus end of astral microtubules to actin cytoskeleton of cortex. It pulls spindle poles towards the cell cortex and away from each other. Depolymerization at plus end pulls kinetochore towards pole is helped by kinesin 13 and kinesin 7. In anaphase, separase enzyme degrades cohesin. Before anaphase, securin protein keeps separase inactive. Destruction of securin is done by APC/C activated by MCDK. After separation, each chromatids pull towards opposite poles. Anaphase is divided into two stages, anaphase A and anaphase B. Anaphase A is initial poleward movement helped by kinesin 13 and 7, shortening of microtubules by losing plus end microtubules. Anaphase B depends on motor protein at pole and central spindle. Two poles moves farther apart due to sliding of polar microtubules and pulling forces exerted by astral microtubules. In telophase, daughter chromosomes arrived at opposite poles. Kinetochore microtubules disappear. Polar microtubule elongate more. New nuclear envelope reforms and nucleolus reappear. Cytokinesis is the division of cytoplasm that begins in anaphase and ends in telophase. In animal cell, it is done by cleavage by contraction of contractile ring made of actin filaments and myosin II bound to cytoplasmic face of plasma membrane. It makes cleavage furrow. In plant cell, it is done by cell plate formation. Formation of phragmoplast, array of microtubule with actin filaments assemble and form like cylindrical structure. Small vesicles from Golgi body filled with cell wall precursor fuse with phragmoplast and form early cell plate. Later cellulose microfibrils are laid down within cell plate to complete cell wall. In meiosis cell division, chromosome number reduced to half to 4 daughter cells. It is divided into two parts that are meiosis I and meiosis II. In meiosis I, prophase I is divided into 5 subtypes: leptotene, zygotene, pachytene, diplotene, and diakinesis. First, let's leptotene. Two sister chromatids appears to be single chromosome ends attached to inner nuclear envelope. There is bouquet formation in which tips of chromosome move until most end up in limited region near each other. It facilitates pairing and synapsis of homologous chromosomes. Next is zygotene. In zygotene, synapsis initiated. Synapsis starts when homologous ends of two chromosomes are brought together on nuclear envelope and proceed inward in a zipper-like manner from both ends. Or synapsis begin at internal region and proceed toward ends. After synapsis, pair of homologous chromosome is called bivalent or tetrad. Crossing over occurs in pachytene. It is reciprocal exchange of equal and corresponding segment between non-sister chromatids in pair homologous chromosomes. Higher crossing over frequencies is called hot spots and lower crossing over frequencies, cold spots. Desynapsis begins in diplotene. Synaptonemal complex dissolves, allowing two homologous chromosome in bivalent pulls away from each other. Each bivalent remain joined by one or more chiasmata. It is site where crossing over takes place. Chromosome decadence and synthesis RNA. In some case, highly active in RNA synthesis results in lampbrush chromosome in amphibians or other organisms. RNA synthesis cease in diakinesis. Chromosome condense, thicken and become detached from nuclear envelope. Then, metaphase I. Here there is breakdown of nuclear membrane, attachment of kinetochore microtubule to bivalent, homologous pair moves to metaphase plate, independent assortment of chromosome along metaphase plate. In anaphase I, homologous chromosomes are separated. Sister chromatids stay together and inhibited their separation by special meiosis-specific cohesin, Rec8, chromosome numbers reduced to half, hence it is called reduction division. Chromosomes arrives at poles in telophase I. New nuclear membrane surround each haploid set of chromosomes. Chromosome uncoiled to form chromatin. Cytokinesis occurs and produce two daughter cells. The gap between meiosis I and II is called interkinesis. In meiosis II, there is no DNA replication. It is similar to mitosis.